Welcome to the Chapter 5, Part 2 lecture on proteins. You should use the information in this lecture to complete the Chapter 5, Part 2 guided notes, which of course you should complete before you attend class. In this lecture, we're going to talk all about proteins. We're going to talk about what they do, how they're put together, and how they're folded into a three-dimensional shape. We'll also discuss how things like mutations can affect the shapes and functions of proteins. So let's get started. So, what are proteins and what do they do? Well, most people know that proteins are things that you get in your diet from eating things like meats and fish, eggs, and legumes. And they know that they're good for building muscle, but not really anything beyond that. Well, this is what proteins look like in reality. Proteins are big, complex molecules made of hundreds or even thousands of little subunits. They have many different shapes and sizes. There are hundreds of different proteins inside your cells, and they all have different jobs. So proteins are complicated. If you've ever wondered what your DNA actually does, well, what your DNA does is it actually holds the code for making proteins. So the DNA stores the information for making these molecules, and then these molecules actually do all the work of the cell. So by controlling the DNA, really you're controlling the proteins. So proteins are extremely abundant in every single type of cell. If you took a cell and you dried it out, you dehydrated it, you'd find that 50% of the dry stuff that was left over was made only of protein. What kind of things do proteins do? Well, they do all of these things. They do lots of jobs. Again, they run the cell. So if you imagine a cell as being like a little city, the proteins are like the little people inside that do all the little jobs. So let's go through some of these. This diagram comes from your textbook and it illustrates just a few of the functions that proteins perform. One of the most important types of proteins in your cell are these things called enzymes. The enzyme is this purple blobby thing, that's its particular shape, and you can see that what it's doing is it's grabbing this dimer and it's breaking it down into individual monomers. So enzymes control chemical reactions inside your cells. They break things down or they build things, and they control the rate at which these things happen. There are many different enzymes inside your cells, and they all have slightly different jobs. We'll talk about them more over in Chapter 8. Some proteins have a protective function. These proteins are called antibodies, and notice that they're shaped like little Ys, which is kind of interesting. But what these things do is they stick to the outside of invading particles like viruses and bacteria, and they help prevent them from spreading. So that's an immune protein. Some proteins act as storehouses for building other proteins. A great example of this is egg white. Egg white consists of a protein known as albumin, and albumin can be broken down into its monomers, which are called amino acids, and that can be used to build other proteins. So you find this albumin inside bird eggs, like chicken eggs, and it's there as a storehouse to build proteins for the little chick that's growing inside that egg. Another type of protein is known as a transport protein. We're going to talk about this in more detail in Chapter 7. Transport proteins are gates. They're built into plasma membranes, the cell membranes of cells, and they help control what goes in and what comes out of the cells. So they're the gatekeepers of the cell. Here are some more functions of proteins. Some proteins help your cells communicate with one another. These include things like hormones and things called receptors. Hormones are proteins that are released in the blood, and they circulate around in your body until they reach what are called target cells. Then they bind with those target cells and they send messages. A great example of a protein hormone is insulin, which is released by the pancreas, and it talks to the liver and your muscles and tells it to remove sugar from the blood. Now, this system wouldn't work without receptors. Receptors are little proteins built into the membranes of cells, and they will connect with the messages, and that causes a chain reaction to happen inside the cell. So they're like the mailboxes that receive the message. Finally, we have two examples here of structural proteins. Some of the most famous structural proteins are found inside your muscles. They're known as actin and myosin. If you could see inside your muscle cells, you'd see that they are just filled with these little proteins, and what these proteins do is actually grab each other. When your muscle contracts, these guys grab each other and pull, and that's what allows your muscle cells to contract and get shorter, and that's what allows you to apply force to something. Here's another example of a structural protein known as collagen. Now, you may have heard of collagen in skin products. Lots of skin products claim to have collagen in them or help build collagen. Look at this protein. It's shaped like a bundle of springs, and being stretchy is essentially its job. Collagen is found deep inside the skin, and it's the protein that allows your skin to stretch and pop back into place. Now, as we get older, our collagen begins to break down, and we don't make as much of it anymore to replace it, so our skin begins to sag and develop wrinkles, things like that, and that's why you see it in a lot of beauty products. Now, beauty products that claim to grow collagen, those might work, depending on what's in there. Beauty products that claim to contain collagen, on the other hand, that's probably not going to help you at all because collagen is a big protein and it can't soak into your skin the way a little bitty molecule can. Right? But those are some functions of proteins. Like all biomolecules, proteins have monomers and dimers that are specific to them. In the protein group, the monomer is known as an amino acid. Here we see two amino acids being put together into the dimer. The dimer of the group is known as a dipeptide, and these amino acids are held together by a peptide bond. Anytime you see that term pep, I want you to think protein, because pep often refers to protein. Now, before you leave the slide, I want you to think, what kind of a reaction is this that I'm seeing? I've got two small subunits being put together into one large subunit, and look what's coming out. What kind of reaction is that? You know this. The polymers in this group all consist of long strings of amino acids held together by peptide bonds. There are two different kinds of polymers that can exist, and these really are put into these categories based on their function. Proteins are fully functional, so they can go out and do a job all by themselves. Polypeptides, on the other hand, are not fully functional by themselves. They have to be combined into larger machines in order to perform their function, in order to do their jobs. Again, the monomers of this group are known as amino acids. There are actually 20 different amino acids that exist. Their names end in "ene." So this, for instance, is tyrosine, this is asparagine, this is valine. One of the skills you will practice in class, and you will be tested on later, is labeling the parts of an amino acid. So let's take a look at it. Even though there are 20 different amino acids, they all have certain parts in common. At the heart of an amino acid, we have this carbon that we call the alpha carbon. Now, because carbon can make 4 covalent bonds, there are going to be 4 things attached to it. All amino acids are going to have a carboxyl group on one side and an amine group on the other side. This is where it gets its name, amino acid. Remember that carboxyls are acids. You will also find a hydrogen attached to that alpha carbon. It's kind of there just as a space filler. And then finally, you'll have something known as the R group. Now remember that R in chemistry is a wild card. It means that there's something attached there, but we don't know what. All 20 of the amino acids have the amine group, the carboxyl group, and that hydrogen around the alpha carbon. So that doesn't differ. What differs among the amino acids are the R groups. So let's take a closer look at those. Let's practice labeling the parts of some amino acids. At the center, I've already identified the central alpha carbon. It's circled in red. Now attached to the alpha carbon, you're going to find the other groups. So here we have the carboxyl. Here's a carboxyl. Here's a carboxyl. Now that carboxyl may look a little funny to you, and that's probably because it has a negative symbol here instead of an H. Remember that carboxyl groups are acids, so what this molecule has done is it's ditched its hydrogen. It's put it out there, which is why it's classified as an acid. Next, we need to identify the amine groups. Amines are NH2 or NH3. In this case, they are NH3's pluses, and here they are in blue. Now amines act as bases, and so that's why they have taken that hydrogen out of solution and attached it to themselves. Now what's left over? Well, we have the R group. Everything from the alpha carbon down is going to be part of the R group. Notice that the R groups of these three amino acids are really different from one another. This is what gives them their different properties. The 20 different amino acids are classified in three categories. Some of them are nonpolar or hydrophobic, some are polar or hydrophilic, and some are electrically charged, and they can be broken down into either acids or bases. Let's take a look at them. Here we have the 20 different amino acids in their three categories. This top category is known as the nonpolar group. So why is that? Well, if you look at the tip end of the R groups of these amino acids, you'll see that they end in nonpolar functional groups, such as methyls or carbon rings. These amino acids do not like to interact with water. Here we have the polar group. Notice that the ends of these end in hydroxyls and things that like to interact with water. They are hydrophilic because water is polar and is hydrophilic. Here we have the acids and bases. Notice that the ones that are classified as acids end in carboxyls, which is an acid. The ones that are classified as bases end in amines, which are basic. Now, why do we care about these categories? Well, we're going to use these amino acids to build a larger protein. And when that protein begins to take on its shape, certain amino acids will only interact with certain other amino acids. So the amino acids that are used to build the protein are going to affect the shape that it eventually takes on. In general, nonpolars only like to hang out with nonpolars. They do not like charges. Polars, on the other hand, will bond with polars. They will sometimes bond with these electrically charged guys. And the electrically charged guys will often make bonds with any amino acid that has an opposite charge.